Good afternoon, everybody, and welcome to the next in the series of British Society for Immunology coronavirus webinars. It's fantastic to have you all here again. We have hundreds of you watching this webinar live already, over 500 people logged in, and that number is going up considerably and quickly, which is fantastic. And we've got people from across the world joining us, so welcome to our UK viewers, but also to our overseas viewers as well. Just as a reminder, the British Society for Immunology is providing these webinars in association with our two journals, Immunology and Clinical and Experimental Immunology, but we're also doing a whole heap of other things in the coronavirus and COVID space. So please do visit our website, our Connect on Coronavirus Hub, which has got lots of resources on there, recordings of past webinars, and lots of information that you will all find very useful at this time. So please do visit that website. Today, though, it's my absolute pleasure to be introducing our speaker. It's the wonderful Professor Paul Klenerman from the University of Oxford, and today he will be talking to us about some of the great research that he's doing at the moment, but also previously and research from across the community, looking at coronavirus, looking at COVID, and also what we can learn from other infections and other viruses that have been affecting us over the years. So we'll learn a huge amount from Paul, and you will also have a chance to ask questions at the end. So as usual, Paul will speak for around about 30 minutes, and then we'll have 10 or 15 minutes for questions at the end. So please do submit your questions via the questions tool on this platform, and I'll ask as many of those questions as possible of Paul before we finish at 1:15 this afternoon. So I will leave it there. It's time to hear from our wonderful speaker. Paul, it is over to you.
Great, thanks. So I'll just try and get the screen up. Yeah. Well, thanks for inviting me to talk. So I'm going to talk about some of the work that's going on in Oxford, just to give a bit of context. And I'm just going to pick out a couple of things that I find particularly interesting based on what we've done before. So I'm sort of presenting in that context on behalf of a lot of people here, which we've organized ourselves into the Immunology Network with the help of the BSI, and these are some of the different units involved. By no means all of them. There are a large number of people contributing to the effort. So, I mean, I guess the question that we're all addressing as immunologists is related to this, which is everybody can get exposed to SARS-CoV if it's circulating. But some people end up with very mild disease or almost an asymptomatic illness, and some people end up at the other end of the spectrum with severe lung injury and death. And we understand some of the kind of demographics that are associated with that, age and sex, and increasingly some idea about ethnicity and underlying diseases. But ultimately, it's the role of the immune response that's going to be critical in determining these outcomes for the individual, and then of course that magnifies as to an impact on populations. So I'm sure everybody watching this doesn't really need any kind of immunology revisions, but I'll quickly run through these slides just to orient people about what I'm going to talk about. So I think for this, and of course many other virus infections, the very earliest responses are going to be critical. So things around the mucosal defense, particularly interferon responses, and in the context of the lung, lambda interferons are known to be important, and there's already some data emerging on that in the gut as well for this SARS-CoV-2. So that's one set of very early mediators which set the scene. The lambdas are much more local than the alphas, which will create a systemic response. And I've just flagged up on there, of course, they're local T cells subsets, and I'm going to talk about some of those later, which are interfacing with myeloid cells to provide very early aspects of the host defense. So that's that. And then, of course, a lot of the work that I've been involved with has been around the more conventional T cell response. That takes clearly a longer to generate because it's got to go through this loop through antigen presenting cells back to the lymph node to create the effector populations. What's not on this diagram are all the resident memory populations that have been laid down by previous coronavirus infections we've all had, and to what extent those are cross-reactive. That bit is really fascinating, but I haven't got any real data to show on that. That's obviously an area that needs to be worked on. And at the moment, I think people are still neutral about the relative roles of CD8 T cells and CD4 T cells, although the early studies seem to suggest the things you can measure in blood are somewhat biased towards a CD4 response. But it's still relatively early in the understanding of that. And I haven't got a slide on B cells, but obviously that's a critical component to this too. So the other thing I wanted to kind of just get across at this point was in any viral infection, there's a balance to be struck between immunity in terms of clearance of the infected cells and immune-mediated pathology that leads to inflammation and tissue damage and dysfunction. And that's quite finely set, actually. The things that impact on that are both factors in the host. Clearly the genetics, sex and age, they all play a role. Some of these are much better understood than others. And then viral factors, so you can make small mutations in viruses in different models that have massive impacts on the outcome, as well as the dose and the route of infection. And this is clearly going to be important in COVID-19, but it's important in every virus infection. So if you look at studies of all the viruses I've listed there, there's a balance between host immunity and host immunopathology. And that includes studies, I guess, the closely related ones include influenza, which I'll describe a little bit later, the original SARS virus, and then the classic LCMV, lymphocytic choromeningitis model that has been studied for years, where you basically all the pathology there is driven by the immune response, and it's been very useful in trying to tease that apart. So that's a kind of background. And whether a response is protective or not is very often not about the quality of the response, but when the response is elicited and how long it's sustained for. So people often ask, is this a good or a bad response? And the answer is always, it really depends when. So this is just a slide about what we've tried to organize ourselves into in Oxford, because there were lots of people in the network, and we tried to make sure that things were reasonably streamlined. So we've got 4 kind of main work packages based around patient sampling. Most of this is through local patients coming to our own hospital, the John Radcliffe. We're also linked in with ISARIC, which Peter Openshaw, I know, discussed, and is linked in with Edinburgh and Liverpool and many other centres. So that it's a kind of part of a really good national effort. And we've got our own cohort of healthcare workers that we're now studying, which is accumulated to 10,000 have been screened so far. So there's some very large cohorts emerging. So we started off with a package on humoral immunity, which is led by Gavin Screeton, developing some antibody tests, some of which have been already published, that in all with the mix of other antibody tests, and developing some therapeutic monoclonals, and just exploring the quality of the B cell responses as well as serosurveys. We've got a big piece of work using genomics to try and pick apart the pathology by comparing patients with different clinical courses. So there are about 140 samples going through a single cell or site-seq program at the moment. So that's quite a lot of data that we're going to be generating, which we'll make available to everybody. I think that's one of the really important things, is to try and create these data sets for the general good. And that's backed up by similar sorts of studies using flow and so forth. There's an important piece on the innate immune response, which I'll just describe some of that, but I think a particular interest that's emerging is how that interfaces with the coagulation cascade and this very strange clotting issue that's important for patient presentation and quite hard to treat. And then there's a lot of work on what the T cell response is doing either good or bad, how this relates to disease outcome, how it might relate to vaccines and protection. And we're linked in with the vaccine studies that are going on locally using adenoviral vectors, and we're trying to apply all this immunology to therapeutic trials, particularly things like anti-TNF and the REMAP-CAP study of antibody therapy. And then, as I mentioned, we're trying to get this data together in a usable format and get it out to probably most of the people watching this webcast will be interested in some aspects. So that's the ethos anyway. I hope it works. And although I'm going to talk about a couple of things in particular because they're close to my heart, I'm happy to answer questions about any of this stuff, or at least I'm happy to be asked the questions. I'm not sure whether I'll be able to answer all of them. So I'm going to just describe a couple of things based on this idea of the timing being really important and getting stuff going early. I'm going to talk about some work that we've been doing on the interferon responses, or at least the regulation of ACE2 by interferon responses. And I'm just going to describe mate cells because I think they might be relevant to this and many other viral infections. There isn't so much new data there, but hopefully the story is reasonably relevant. So I'll dive straight in then with kind of some data. I'll just move something on my screen just to make it a bit easy for me to present. So what we did was we took a look at some RNA-seq data that we generated recently from a study of hepatitis C. And you might say, oh, well, this has got nothing to do with SARS-CoV-2. But essentially, we were interested in innate responses, and they're both RNA viruses, and many of the interferon responses overlap substantially between different viruses, even in different tissues. I mean, they're obviously important differences, but we're really asking the question, what is regulating ACE2 expression in the context of a virus infection? And while we'd love to answer that in SARS-CoV-2, that's going to take a little work to do. So ACE2 is important, of course, for a couple of reasons, but most people will know it's the major receptor for SARS-CoV-2 as it was for SARS, and it's got an important physiological role as well, which we often neglect as immunologists. Well, at least I had, certainly. I suspect many people had kind of not really considered this while we're thinking about the next T cell or cytokine. So what you can all see from these graphs, I hope, is that there's an inverse relationship between the expression of some of these genes, IFIT-M1, IFI6, BST2, and RF7. We just picked 4 to illustrate with ACE2 expression in these liver biopsies. It's about 160 liver biopsies that we've taken from patients who are going into a clinical study. It's a very, for those of you who don't do this kind of thing, it's a very large study of that from that point of view. And we've also sequenced the virus in all of them from end to end, and we've SNP typed the individuals from end to end. So it's a sort of genome to genome study of virus infection. And what I hope you can see on the right is the aggregate association. So this is the cells which these are the pathways which inversely associate with ACE2. So all of the top ones are basically overlapping versions of gene sets which you can informatically pull out, which are inversely related. In other words, they show the same pattern as I've just put up on the slide here. The more interferon you have, the less ACE2 you have. You can't tell from this exactly which interferon. It says type 1, but the type 3 interferons and to some extent gamma have very overlapping gene enrichment patterns. So it's essentially interferon. I think you can say pretty conclusively from this, there's a very high degree of certainty in these analyses because you pile up all the genes and you get a lot more power. Many of you who are interested in this might say, well, hang on, isn't ACE2 an upregulated gene by interferon? Because there's a Cell paper that's just been published and it was around in preprint for a while. But I think the difference is that well, as far as I understand it, that paper was really looking as an in vitro response to interferon, which is a very classical way of looking for RSGs. And this data is all in vivo, and maybe the context of the interferon is rather important. It's also true that in this study, at least, that the interferon is chronically stimulated, so that might be another important difference. But anyway, it does look pretty convincing to me, and hopefully to you. Now, you might say, well, this has got absolutely nothing to do with coronaviruses. It's Hep C in a liver. So, and you wouldn't be alone in thinking that, but we repeated the analysis twice actually in different human gut cohorts. So these are patients with different sorts of inflammatory bowel disease of different levels of inflammation, and we picked in first case people who hadn't had any therapy at all, and in the second case we kind of enriched for people with different interferon genotypes. But in both cases you've got the same result, that in the gut, this is a small bowel, you get an inverse relationship, but these are the same genes basically, and with the same interferon signal inversely associates with ACE2, slightly different levels of expression, but exactly the same relationship. And then I think what's really interesting is if you look in mouse studies, this is getting to the lung, and this is a study that was published on influenza from the Penninger group, who's very interested in ACE2, and they found downregulation of ACE2 as interferons were upregulated in mouse lung. And he's gone on to knock out ACE2 and show that, in fact, in those mice, there was a significant increase in pathology when you lose ACE2 entirely. So although it's downregulated physiologically, there is a requirement for ACE2, and essentially the pathology was mediated by angiotensin, and he could block that in that model. It's quite a nice set of papers. So it did look pretty convincing across tissues and species that you get this same relationship. So you can start to probe that a little bit more in our study and say, well, why do different people have different levels of interferon and therefore ACE2? And because we typed all the individuals with SNPs, you can see that there's a set of polymorphisms here that link to ACE. This is essentially a Manhattan plot for ACE2 expression. So that this inverse relationship is driven by this locus here, and that locus is interferon lambdas. So lambda 3, 4 are situated there in the human. And this is an area that people interested in Hep C have known about for a while. It's a very, very powerful impact on clinical outcomes, both spontaneously and in response to interferon therapy. So what you see if you do the sort of analysis then is that the impact is driven by the genotype, and we split this into what's called CC and non-CC genotypes. So lambda 4 is a gene that's only present in the non-CC genotype. It's a complex bit of genetics, and I don't really have time to go into it, but you just have to trust me that these guys make lambda 4 and these guys don't through a phase shift. And actually lambda 4 was only really discovered through very, very careful analysis of these sequences. It's very hard to measure any of this. It's all been done through informatics, and some pretty careful studies have confirmed this. So it definitely agreed that there's a lambda 4 positive and negative group. Age also has an effect, cirrhosis has an effect. So all these cases have higher levels of ACE2. So these guys with a non-CC genotype have higher levels of interferon. They continuously stimulated to generate interferons. They're less susceptible to interferon treatment because they already got their interferon system on maximally. Age also has an effect, as you can see here, and sex, actually it does have an effect, but you have to kind of pick things apart very carefully to see what that is, and it's not so obvious on ACE2, although it depends how you do the analysis. Certainly sex has quite a big impact on the overall interferon signaling. And what's interesting about this gene locus rather, because it's a set of linked genes basically, this SNP is just giving you the overall picture, is that it's quite differently distributed in different populations. So the CC genotype, it's probably easier to look at this graph. The CC genotype, which is they don't make any lambda 4, is a sort of more of an East Asian genotype. There's very little of the TT genotype, which makes only the lambda 4. That's much more of a specialty of the African, particularly the West African, and also the African American population. So the regulation of ACE2, at least in context of Hep C, is quite different between these groups, and it's possible, but of course we don't know, that this would be true for other viruses. This particular genotyping has been done in a set of African kids, looking at respiratory viruses, and they found that there was an impact of which of the genotypes you had on clinical outcome, and it was a mixed set of respiratory virus infections, and not just coronaviruses. So there is some association that was a clinically kind of based study, and the mechanism wasn't clear, but it's certainly relevant to respiratory infections. And the sort of model is that, well, at least in my head, is that in the steady state in a given tissue, there's a certain amount of ACE2, and then when the interferons come along, you downregulate ACE2 as the interferon gene kicks in over a certain time course, which needs to be explored. And with lambda4, this signaling is somehow sustained, and it's a really interesting phenomenon, and it's seen not just in virus infections but also in pregnancy, that the lambda4 genotype is just sustained and comes off much more slowly, and so the level of ACE2 will remain lower. In the case of the CC genotype, it's possible that the early events of induction of the whole interferon response, and therefore the downregulation of ACE2, might be somewhat earlier. It's a little bit hard to pick out from the data, but in Hep C, this is a response that is associated with more rapid clearance of virus. It's quite a big effect, about threefold. Okay, so I've spent a lot of time on that. I'm going to quickly whiz through mate cells. So these haven't been formally linked to COVID yet, but I'll show you a little bit of data on them, and just to get everybody up to speed, and hopefully at least everybody at the end will know how interesting they are. This is a group of cells which you'd be forgiven for overlooking in the past because they had a bit of an obscure sort of birth as just a type of invariant TCR that you could pick out. Subsequently, it was shown that this TCR is restricted, in other words, recognizes MR1, which is an incredibly conserved MHC-like molecule, which got a groove I'll show you in a minute. It's fairly non-polymorphic, so that humans and mice can effectively cross-recognize MR1. And subsequently, it's been shown they express CD161. So if we were to bleed members of this webinar, we would always find a population of cells which high expressors of CD161 and the alpha 7.2 circulating in blood, and it's quite variable, but it can easily be 10% of your CD8 cells, and it's quite restricted to this very bright group. CD161 is equivalent genomically, at least, to a kind of NK1.1 locus in mice. It's an NK marker, a C-type lectin-like molecule. Effectively for this, it's just a good marker of the cells. If you use a tetramer, which is MR1 with a ligand in it, I'll describe that in a second, you get exactly the same set of cells. And if you look in the liver, and we haven't done this so much in the human lung, but we're getting there, these populations can be huge, about 30% of your CD8 cells, and a very good fraction of overall T cells. So these cells bind a riboflavin intermediate that's made by bacteria, or at least they recognize, their TCR recognizes that riboflavin intermediate expressed on MR1. They express raw gamma T, they make R17, they look antimicrobial, and indeed they are, because if you offer them E. coli, they will respond by making interferon gamma. This again is just straight out of the blood of a healthy donor. This is a crystal structure of the MR1 with this is not the ligand actually, it's what was originally crystallized, which is a folate derivative in the group, but it's very small molecules that are on the way to making riboflavin. And then they also express a lot of things that might be antiviral, including this transcription factor PLZF. And we, in the past, I'll run through this fairly quickly, but we've tested a bunch of viruses to see whether they induce mate cells based on this idea that they might respond. And indeed, these are clinical data from a while back, but they've been reproduced by many, many groups in different viruses. And you can see in all cases, you get this upregulation of granzyme B, which is you could use any marker, but granzyme B is a really good one because in healthy donors, you don't see much. In acute dengue or acute Hep C or even chronic Hep C or acute severe flu, you get the same thing happening. And here's the COVID data. So this is preprint that shows, again, in COVID, there's activation. They've used CD69 in this case and PD1 to show that the mate cells are activated. They also managed to look at some iNKT cells. So in humans, they're about the ratio of iNKTs, mates to iNKTs is about 100 to 1. These are much rarer than mates. In mice, it's the other way around. There are many more iNKTs than mates. But they kind of fill a similar ecological niche, and that they will respond in parallel in many cases. And there's subsets of gamma delta cells, particularly the delta 2s, that will do the same thing. So we know they're activated now in COVID, and the question is really what they do. And people just might be a bit confused why a T cell will be activated in a TCR independent way. But if you block MR1, this blocks antimicrobial function, or at least recognition through the TCR. But it doesn't do anything here. This is all mediated by cytokines in IL-18, IL-12, but also IL-15, type 1 interferon, and even TNF play a role. But in the dengue model, for example, if you block IL-12 and IL-18, you kill off the response. So it's effectively like an NK cell. It's an innate function. So they're kind of a bit kind of split personality. They'll either work as a conventional T cell or as an NK cell. So we've tested the role of NK cells in vivo using mice. And this is experiments we did a couple of years ago in Australia with Tim Hinks, and we had to go to Australia because they had everything really well set up there. So we used their setup, and the body Van Wilgenberg went from my lab and worked with Jim McCluskey, and so this was published. So again, I'll run through it quickly, but I think there's an important message that I just wanted to explore with people. So first up, the cells expand pretty quickly in the lung compared to, this is all fold change. So that in the mouse lung, they're enriched compared to other organs, but they're still very infrequent compared to humans. And I wasn't sure this experiment would work at all, but it's worked well. But they expand prior to that enrichment of other conventional T cells coming into the lung. So it's early. And it doesn't matter what strain you use, but the X31 strain's a kind of conventional flu strain. And I can just say right up now that it didn't really make any difference what we did with the mate cells. They all activated, but they weren't necessary for protection. Whereas with the PR8 strain, we got a different story, which is a much more severe flu. The weight loss was more significant. And the differences are relatively small, but they're quite big experiments in the end. And the weight loss in these mice is in a wild type mouse, if you set it up right, you can get a significant weight loss that they'll recover from. But the MR1 knockouts, if you look at this, they tended to not recover through that body weight loss. And if you transfer back in mate cells, so MR1 knockout mouse doesn't have MR1 in the thymus, so it can't generate the mate cells, and they are deficient. So if you replace those cells are working in an MR1 independent way, and you can recover some of that. So it just suggests that having mate cells around can protect you against death from a severe viral infection. So it seems to me to have some implications for COVID, even though we still don't yet know the story. What the mate cells are doing here isn't yet understood, but it looks to me like they're accelerating the accumulation of other cells. So macrophages have to come in, and of course, and in the case of a mate cell deficient mouse, they just come in later. And the wild type mice, as I showed you, recruit T cells, and that's all very blunted. So the whole thing is essentially slowed down in the absence of mate cells. It's not like it doesn't happen, it just doesn't happen at the right time. And it's possible that these very early cytokines, the mate cells essentially take early interferon signals and amplify them, and then they will recruit other cells very efficiently. So the MR1 knockout mice just lack some of the early, for example, MCP1 signaling. So in this model, which is, it says flu, but I think it would be relevant to COVID, in this sort of loop of activation and then recruitment of cells that allow for clearance, it looks like mate cells are acting relatively early, and other experiments that have been done suggest it's through interferon gamma, and just accelerating this loop. But what's really interesting is that this paper and many others have suggested that mate cells are kind of lost in the elderly. It's pretty profound, actually. Once you get into your 80s, it's quite hard to find. Of course, all these aging phenotypes are very variable, and mate cells themselves are very variable even in healthy donors. If you take just average lab donors, it's huge differences, but the trend is undeniable and is pretty consistent across studies. So it's likely that that's playing some kind of a role. Whether it's an important one or not, I think needs to be really defined. And I just thought, since I've got the floor for another minute or two, I'd just show you my postdoc Nick Praveen's data about vaccines. And this is kind of relevant because one of the vaccines that's been used is a vectored vaccine expressing spike on this backbone ChAdOx1. So that these are all adenoviral vectors. So they're vectors which are disabled based on adenoviruses, and all of the adenoviruses to varying degrees activate innate immunity and will trigger mate cells. So they're very good platforms in inducing T cells generally, and we've been exploring the role of mate cells in this. So in this trial, which was set up very nicely by Andy Pollard for us, if you look at very early time points, the mate cells get strongly activated, and that correlates with the degree of, or the levels of circulating cytokines that you get at that point. And it's the same in mice. The mate cells will be activated by these different adenodes. And interestingly, AD5 doesn't generate quite the same levels of cytokine response. It's more of a stealth adenode. ChAdOx1 is in a different clade of adenodes, and it generates a lot more IL-18 and interferon. So that is the kind of human data, but then you're still asking the question, well, so what? I mean, the cells get activated, do they do anything? But actually, since you've got the mouse model, if you knock them out, this is sort of data using a hep C. Again, it's ChAdOx1, but it's got a set of hep C peptides in it, or it's a string of antigen which create the peptides, and we're using the peptides to test the response. So these are no peptide, and this is a response to the vaccine in a wild type mouse. You can see these cells are vaccine induced by this ChAdOx vaccine, and in the same strain. So they're just missing mate cells, and the mice are otherwise fine, and the mate cells are a tiny fraction of T cells. There's actually a very profound effect on just the immunogenicity, and of course we're studying this with the ChAdOx spike as well, but it seems to be that essentially we've tested it in a few different vectors, and we get the same reproducible impact of taking away mate cells. So they're not only protecting your respiratory tract, but they're probably having some impact in response to vaccination, at least with these virally vector vaccines. So that work is still ongoing. So I think I'll stop there. It's about time, and I'll thank everybody. Azim Ansari did all the analysis for the Lambda stuff. As I mentioned, Nick Praveen has done all the work for the mate cell stuff, and then hopefully I mentioned a few people as I went through for the Oxford team work. And yeah, happy to answer any questions now.
Brilliant. Thank you very much, Paul. That was a fantastic presentation, and it kept the crowd, so everyone stayed on, you'll be pleased to hear. And we had a huge number of questions through, so again, apologies to viewers if we don't get a chance to ask your question of Paul, but we will ask as many as we can between now and 1:15. So Paul, I tried to group the questions in different themes, and the first one was really from something you mentioned at the beginning of your talk around CD4, CD8 T cells and the balance of those that we're seeing in COVID patients. Obviously, classically, CD8 T cells are involved in antiviral immunity. What role do you think there could be for the CD4 T cells?
Yeah, well, so I think CD4s often get neglected in the antiviral responses, so they're really critical in providing early help. If you take those away, you can generate a CD8 response, but it's not very effective and it's not very sustained. They can have their own effector functions, of course. Cytolysis is probably not so critical on average, although there are some examples where cytotoxic CD4s are important. And you tend to find that more in tissue. I wouldn't imagine that's the dominant thing. And then there's a kind of unanswered question about follicular help and the generation of a really good antibody response, a really high quality one. So all of those things are going to kind of combine to to provide a really important role for CD4s. It's interesting that they, normally when we look at these responses, the CD8s really leap out at you, and the CD4s are kind of buried under a big pile of gamma secretion from the CD8s, and it doesn't quite look like that with COVID, and it may be something to do with the kind of duration and the timing that we're looking at, I mean, and it's also possible, of course, that the virus is doing something to kind of limit some of the presentation to CD8s. So it's not completely out of sync with what I've been told some of the responses in the Flu Watch study look like when you unpick the very big study of influenza that Andrew McMichael and others around Oxford Imperial have been doing, Anjit Lalvani as well. So I think that there is a sort of CD4 to CD8 are more normalized than what we perhaps see with some of the really strong virus infections like HIV and even acute Hep C, where there's a sort of massive response.
Great, thank you. And as you can imagine, lots of questions about different cell types coming in and what we know about those in terms of RNA viral infections, but also COVID-19. So I'm going to fire a couple at you, Paul, and if you could give us your best answer to them, that'd be great. So regulatory T cells is a question mark. Do they have a role to play? Does it look like they're having a role in this disease? And dendritic cells, are they important in the infection management or the disease?
Yeah, that's a good question. So the Tregs, I haven't seen anything published yet, and I think that will be uncovered, because essentially what's needed to do is unpick the phenotype of those CD4s that people can measure now and see. I mean, obviously some of them making gamma are unlikely to be Tregs, but the other thing to say about that question is probably the place to look is in the respiratory tract. So there are studies with Imperial and elsewhere going on in the nose and in the airway fluid to see what's happening. I mean, I guess the question could be, I mean, if you have appropriate regulation, do you have a relatively mild course, and then is some of the very severe disease that looks like it's immunologically driven associated with a lack of regulation? I guess that would be the hypothesis. I really don't know the answer. There's a paper on circulating cytokines that flashed up in the Lancet recently, and I'm not sure if that answers the question, but there is some IL-10 measured, so whether that's from Tregs or elsewhere, I don't know. And dendritic cells are definitely going to play a critical role, because if you go back to this hypothesis that if you time it all right and early and get things in the first 48, 72 hours, that the DCs are clearly really critical there. Whether DC, I guess one of the issues then is, how do these DCs or other lookalike cells function in risk groups? So if you look at diabetics, what do we know about that sort of function? We know some of their cellular responses are impaired. Is that at the level of a DC or some other cells? So yeah, really good questions that no answers currently. But what I would say to the audience in general is we will have this very large, dense single cell data set, and obviously other sites will, and people are just welcome to explore their favorite cell when those things come out in hopefully the next few weeks.
Brilliant. And talking of other favorite cells, any comments on the role of NK cells or ILCs? And I mean, you mentioned about the respiratory tract. Do we think they're resident in the lung?
Yeah, well, I'm not sure about the. Yeah, I think there will be. There's that figure I flashed up about where I think mate cells might be fitting in. There's an exactly parallel story for LC1s that came out of Maria Pia, I can't pronounce the surname, but Maria Pia's group in Perth, and she did a very good job with those. And I think there will one of the questions is when you activate a mate cell with cytokines, you should be activating NK cells. And I'm always surprised that we can get these phenotypes on the mate cell deficient mice when there's an NK cell or an iNKT cell that could take up some of the slack. But all these cells are acting essentially in a group, and I think the reason that you might see a given phenotype in a particular organ is just which is the dominant cell type on a given day. So I think NK cells will play a role, and it could be exactly what I just described for mate cells, and it may be that the NK cells are really doing that job in some of the individuals or some subsets of individuals. Again, I think particularly for that one, that we'll generate a single cell data set from BAL, so lung washings, and hopefully people can explore that if those cells are represented there.
That's great. So I think that was it for the PET cells of people out there. Thank you, viewers. A couple of other questions. So there was one around ACE2 in particular, and the question was, given its cardiovascular physiological role, do you think the involvement of ACE2 in the pathogenesis of COVID-19 and the immune response is linked to the vascular shutdown that we see in severe cases?
Well, I think it's really fascinating. So there's something funny going on in the vascular system and the endothelial control of clotting and fibrinolysis that's not very well understood, and it's really tempting to link it through ACE2. And the reason I'm a bit more confident in doing that is that I'd encourage people to read the Joseph Penninger papers, because he did show that when he knocked out ACE2, that there was some of the pathology that he saw in his lungs was driven through excess angiotensin signaling. So this is not an area and it's a very physiological kind of response, and it's a kind of long way from standard sorts of immunology, but it kind of makes ACE2 is able to kind of modulate angiotensin function, and if you lower it, if you knock it out as he did, you could see why you might get a kind of severe phenotype, because you can't really get rid of the signaling, and the way he solved that problem was by blocking it with a pharmacological intervention. So I think it seems to me you could end up in the same place if you just have prolonged interferon lambda signaling and prolonged shutdown of your ACE2. The only bit that many bits I don't understand, but one bit that you could reasonably question is, well, where is that happening, and why would that really impact on an endothelial cell in a given tissue? I mean, the lung is definitely affected by this, but it's not the only organ. So I think that's a gap that needs to be filled. One thing that you might think about is, well, perhaps the virus is just circulating in the bloodstream and it's sort of able to bind these. But when we've looked, and I think the paper is online somewhere, we found, as other groups have found, very little RNA in the bloodstream. The only times we found it was in very, very sick patients. And then when we checked that out, we couldn't grow virus, and it looked like it was fragments of virus that sort of overspill. I mean, it's not to say it hasn't happened at some low level, but it's not a really dominant effect. So I haven't been able to kind of join all the dots, but I think that's the bit that we need to focus on.
That's great. And in interest of time, Paul, because you are a busy person and need to get back to that research, please. Just a final question for you is around the interferon lambda 4. And question is it known that is produced locally in the lung in people who have the SNP that is associated with production? And if that's the case, could it actually be potentially beneficial for respiratory viruses?
Yeah, it's a very good question. So I don't know if it is known, but it's even in the liver, where we kind of spend a lot of time looking for it. It's very difficult to find. And I've not really been able to get my head around this, but it's partly a reagent problem, but probably it's not made at very high levels. And there are some nice papers out there from the Gale group that have looked at the regulation of this. There's no doubt that lambdas are generated. And I think a lot more work needs to be done to look in the very early phases of a viral infection to see whether lambda 4 itself is induced. It's a really good question. So we have set out to try and answer that by taking nasal scrapes and also some biopsies, looking for the transcript if we can find it, or evidence of the transcript in people with the right genotype. So hopefully, if I do this again, I'll be able to answer the question properly, but it's a good question.
That's brilliant. And you would definitely be invited back again, Paul, after that wonderful seminar. Thank you very much. We should bring this to a close now. It is 1:15. So thank you again for everybody joining this afternoon. As always, this has been recorded, and the recording will be available online in the next few days. So please do have a look, and please do let your colleagues know how amazing this webinar was, and for them to watch the recording once it is up there. We do have more to come. Our next one is being given next Wednesday at 3:30 in the afternoon by Professor Judith Brewer, who will tell us about the genomics of SARS-CoV-2 and what that means and what we can learn from the genomics. So please do sign up for that. It's a free webinar, and we look forward to welcoming you there. And don't forget, the British Society for Immunology is here to support all of you at all times, particularly during this challenging time. So please do check out our website, please do get in touch, and let us know if you've got any questions or any other ideas about what the BSI could be delivering for you, our immunology community. So thank you again, everyone, for joining. Thank you so much, Paul, for giving such a wonderful seminar, and we'll let you get back to your important research, and hopefully we'll see you all next week when Judith Brewer is going to be here online. So thank you very much, everyone, and see you soon. Bye.